So when they come together, macrophage is going to say that I have got Salmonella typhi inside me. The T cell will produce interferon gamma. So do you know the phagocytosis process? That phagosome is formed, lysosome is formed, but they will not fuse together to produce phagolysosome until the interferon gamma signal has come. Then they fuse together, phagolysosome will kill the enzymes will produce nitric oxide, and there is whole detail to that as well. So in immune response to bacteria, I'll tell you about those details. So at the moment, this is how things are going to happen. Yes, please.
How do T cells are transported to
thymus? So just couple of slides and we'll get to that point as well. So we'll talk about how they are transported, how they are moving around the body, all these things now. These are all good questions. So the questions you ask, actually it tells you why you have future slides. They are trying to explain some of those points, and sometimes it makes me feel as if you are understanding something as well. So now your next question, it is a very valid question, that why T cell go to the thymus, or how they go to the lymph node, or at what time. So it's a constant process. If you look here, we have got certain, one is the blood lymphocyte pool, one organ here is spleen, this HEV, high endothelial venule, these are basically the veins that allow the cells to enter a lymph node, okay, or a spleen. And then finally at the bottom you can see a box of representing tissues, any tissues, peripheral tissues or whatever. So when the heart pumps the blood, these lymphocytes, they are going to first, some of them are going to go into the spleen. So you can see here, this pool of lymphocyte, this is going into the spleen. So the B cells in the blood, they are going to stay more into the spleen. On the other hand, some blood, it will go into the lymph nodes through, so the arteries, then arterioles, then capillaries, venules, those venules are going to go into the lymph nodes. And then some blood is going into the tissues and become lymph. You know all what is lymph? And then that is collected back into the lymphatics, into the thoracic duct, and into the main circulation. So you can see this last group down, it from the tissues take the lymph, collect it all together, also from the lymph nodes, back through the thoracic duct into the bloodstream. So this circulation keep on happening. So spleen, lymph nodes, blood itself, peripheral tissues, they always will have a circulation of T cells and B cells. And also here, now special thing about thymus is that it is produced, it is, when you are born, it is at maximum activity. Gradually, as you grow older, its mass is reduced and its functional capabilities are reduced. So here in this circulation, it is not mentioned in particular, but as part of this whole circulation, when the blood is pumped, it will also be pumped to the thymus. So especially in the initial years of the life, when you want your T cells to be matured, so that process will take place all the time. So the circulation is happening in all the organs, spleen, lymph node, GIT, respiratory tract, everywhere it is circulating all the time. And there is just one example. There are a lot of things like this. One example is called as CCR7. So it's a chemokine receptor, and it helps the cells to come to the lymph node. So when your body wants that cells come to the lymph node, they start expressing CCR7, and then we call it homing, because lymph node and spleen are two major organs where all this activation is taking place. So homing receptors help the immune cells to go into the spleen or to go into the lymph nodes. That's why we have drawn two small homes here, one on the lymph node, another on the spleen. Spleen is mainly for the B lymphocytes, and the lymph nodes are mainly for the T lymphocytes. High endothelial venules, the small, venules in the lymph nodes. So simply the blood supply to the lymph nodes, at the smaller level. Now we'll look at a small situation that your patient today is a kid playing on a construction site and he is walking along and steps on a rusty nail. As a result of that, he gets a wound and whatever was on that nail is now in our patient. Now, first question is, what happened to the first line of defense? What was the first line of defense? Skin barrier. It is breached. The pathogen has entered. So first line of defense is gone. Now the next thing, do you think B or T cell after release to the peripheral blood from bone marrow or thymus are able to eliminate the infection directly, or do they need to be activated and differentiated to become functional cells? They need activation and differentiation to perform the functions. Okay, now little more into the patient. The patient has got nail penetration at the foot. The pathogens will go, it have two routes. Number one, through the lymphatics, it's going to go into the lymph node. So here is the lymphatic, it will go into the lymph node. Or another is that the dendritic cell, it capture the antigen and take it to the lymph node. So whatever it is, whether it comes through the blood, through the lymphatic, or through the dendritic cell, ultimately the pathogen has now reached the lymph nodes. And so we left one question. So in the beginning we asked, what will happen if there is no thymus?
Suppression of the
So look at the thymus figure here, and what is the main thing that is happening? Excellent. This is the very complete answer, that maturation of T cell will not take place. So if somebody say that there will be no T cell, that's not correct. If somebody say no B cell, that is not correct as well. If you say immune system will be depressed, yes, it will be depressed, but why? Because when T cell don't get matured, they don't come out, they don't get primed, they don't help macrophage, they don't help B cell. So your whole immune system is kind of collapsed. It is not working well. But the main reason is no maturation of T cells. The patient will have T cell, but they will all be immature T lymphocytes. Now, sorry, why do you think lymph node enlarge during infection?
Because of the proliferation.
Excellent. So these cells, these are increasing their army inside the lymph node. So when the size is increased, the lymph node will be palpable. So if you see here, the patient can, so the people who have got sore throat or pharyngitis, their submandibular lymph nodes or cervical lymph nodes, they are palpable, they become enlarged. And the reason is, lot of cells are in the lymph node that are proliferating and they are going to attack the pathogen. So is it a good thing or a bad thing? It's a good thing. Now, why this proliferation take place, or what is mainly responsible for increasing the army of the T cells or the B cell? The answer is interleukin 2. That is mainly responsible for all this. So here this picture tells you that I told you out of 9.1 million, couple are going to recognize Salmonella typhi. Those couple, when recognize it, now they are going to increase their army. So this clone means exactly same, structurally and functionally. They have to be exactly same. So IL-2 can make that by the help of mitosis. So it will tell those cells, special activated cells, to now multiply, increase in number. And this is called as clonal proliferation. Just that clone will proliferate, which can recognize Salmonella typhi. Okay?
Who produces interleukin?
You tell me. Now they are interacting here. Who should produce IL-2?
Thymus?
No. When dendritic cell is interacting with CD4, it is going to activate it. This dendritic cell is going to produce IL-2 and tell T cell to multiply. So this is how they interact, they communicate. So dendritic cell will produce IL-2 to this T cell to multiply. Now in this figure, I've already explained this part down, that in the lymph node, the T cells and the B cell, they multiplied, they become, so one word I missed so far, When these activated T cells come out of the lymph node or the spleen and they are ready to attack, this is called as effector function. So the cells that are ready to perform the function, they are called as effector cells, okay? And once they finish their job, then the remaining few cells are called as memory cells. So now all the different terms that we learned today are immature T cell, naive mature T cell, priming, activation, differentiation, clonal proliferation, effector function, and then memory cells. These are all the new terms that were part of this story today. And then finally, the remaining cells, extra cells, will be eliminated and just the memory cells stay with you. And this is again the same picture to tell you that how cells are circulating in the body. So the pathogen has come down from his foot, while the T cells have come, and the dendritic cell, they have come from the blood. They come into the lymph node, they meet all at the lymph node, and then they are activated and the adaptive immune system starts to act. Now I'll go a little bit in detail about the different subsets of the T cells. So when I was telling you the T cell receptor, I told you T cell receptors are alpha beta if they have two chains, alpha and beta, and they can be gamma delta. Out of 100% of T lymphocyte, 95% are alpha beta, and the remaining 5 to 10% are gamma delta. So the main group is alpha beta, this alpha beta, and the other one is gamma delta based on the T cell receptor. Alpha beta can be further grouped into T helper T cells, and the is called as CD4, cytotoxic T cell, and they have CD8. And then the helper T cell can further differentiate into TH1, TH2, or TH17. Okay, that is based on, or T regulatory cell. T-reg means T regulatory cell. That depends on which type of cytokines come. We'll see that in the next slide. So this is part of the adaptive immunity.
Is the gamma delta the natural killer cell?
No, gamma delta T cell are something different from the natural killer cell. And gamma delta T cell is basically innate immune, like part of the innate immune function. Why? Because they don't have MHC class 1, they don't have MHC class 2, they are not specific. So that's why they are part of the lymphocytes, but functionally they are part of the innate immune system. Now, if you look at the blood cells under the microscope, you can differentiate how neutrophil look, how macrophages, the neutrophils, lymphocytes, and eosinophil look. But can you differentiate between a B and a T cell? No. The only way to do that is either look under the electron microscope, there is certain differences between a B and a T cell, or you look at their markers, the surface markers that they have. So lymphocytes can be B cell, T cell, or natural killer cell. These are the markers that you have to remember. T cell has got CD3, which is part of the T cell receptor complex. B cell have got CD19, and natural killer cell have got CD56. Then, little more, if there is CD8 along with CD3, then they are called as cytotoxic T cell. If it is CD3 plus CD4, helper T cell. If CD3 plus FOXP3, then they are called as regulatory T cell. So this is something that you have to memorize. Now, if you want to ask me this question, that how can you see these receptors? So that is done by a method that is called as immunofluorescent staining, and there is a machine called flow cytometer that can check all this. Now, what is the importance of this? One example I'll give you: the HIV patients, the AIDS patients. At what time, have you ever thought that why human immunodeficiency virus infected patients are called as AIDS patients? So AIDS means AIDS, acquired immunodeficiency syndrome. But any person who is infected with HIV, you should not say that they are AIDS. Why? because there is a certain level. So when their CD4 count, number of helper T cell count, drops below 500 cells, then they are called as AIDS patients. When their cells are above 500 cells per microliter, they are not called as AIDS patient. This is the classification clinically when you will see the patients in clinic. Now, how you will measure those CD4 cells? With the help of flow cytometer. So this is one of the examples. So when you have to monitor an AIDS patient, you have to calculate or measure his CD4 cells and keep doing it when he is on treatment. Every month or after every couple of weeks, when you have to monitor him, take out his blood, send for CD4 counts, and then the number comes, and then you decide, is he improving or is disease becoming more and more worse? Because around 20 million T cells are destroyed sometimes a day in an AIDS patient, and these are all CD4 cells. So this is the importance of why we are telling you all these markers, or why these are grouped like this, because first then there are certain primary immunodeficiency diseases where if cytotoxic T cells are not made, or Dye-George syndrome, for example, how you can confirm it when you will do their flow cytometry. So these are some of the two examples that I gave. In this figure, flow cytometer can see each and every cell labeled. Yes, please.
This is the tough times. Do you have HIV?
Well, if HIV can infect anyone, but those are going to be affected very, very severely. Why? Because first they have very few mature T cells, because their thymus is already absent, and if they are also infected with HIV, it's going to be worse. But how many of them reaches to that age, how many of them get infected, that's a different story altogether. So yes, flow cytometry is the method by which you can measure CD4 and CD8 cells. Why do you think your immune system generate and recall different type of cells during infection? So you have macrophages, dendritic cells, T cells, B cells, antibodies, all of them are participating. Why do you think you need all of them? Do you think only one type of cell will be able to eliminate the infection? No. You need all of them because they have different functions. B cells have something else. They produce antibodies. T cells have got so many types of cytotoxic, T lymphocyte, regulatory T. So all dendritic cells have their own function. Neutrophils have their own function. All cells are performing their own functions. So the simple thing is that you can clap with two hands. It's not possible to do it alone. Now you have all these pathogens. You have got viruses, bacteria, toxins. Some of them are extracellular pathogens outside the cells. Some of them are intracellular pathogens inside the cell. How your T cells know whether it is CD4 or CD8 that deal with intracellular pathogen or extracellular pathogen? How do they know that?
By the receptor.
By the receptor. Excellent. So it's actually, you see, the dendritic cell in the center, they are responsible for deciding. So if a virus comes, with the help of MHC class 2, they are going to present it to CD8. Yes. Yes. Excellent. MHC1 will present the intracellular pathogen with the help of MHC class 1 to CD8 cells. MHC class 2 is going to present extracellular pathogen to the CD4 cells. So here are the main factors that help you decide whether it is intracellular pathogen or extracellular pathogen. And we have already discussed this memorizing trick that MHC class 1 into 8 is equal to 8 and 2 into 4 is equal to 8. Now this activation part, when a dendritic cell come into contact with the T cell, what happens? There are 3 steps involved. Number 1, T cell receptor will encounter MHC class 2, and that is signal number 1. Number 2 are the co-receptors. So beside the T cell receptor, you can see this B7 and CD28. They are the co-stimulatory signal. So number 1 is MHC class 2 and CD4 comes in contact. Then the co-stimulatory signal, they come in contact. The third thing is they produce cytokines. And finally, the type of cytokine is going to decide whether you will have T regulatory cell, whether you will have Th1, Th2 or Th17. I'll just put one cytokine, the most important one I'll tell you. So if you want the Th1, IL-12 is the most important. So if dendritic cell produce IL-12, you will have Th1 cells. If it produce IL-4, you will have Th2 cells. If it produces TGF-beta, it's going to be Th17. And if it produces TGF and IL-2, it's going to be T regulatory cell. So these, if you think based on the pathogen, how the dendritic cell decides, so the brain of the immune system is dendritic cell. That decides that now, with the help of MHC class 2, we want this T regulatory cell, or we want Th1 or Th2. Then they produce that cytokine, and the T cell will convert into Th1 or Th2 or Th17. So it's a polarizing factor. So the three steps are T cell receptor and MHC encounter, co-stimulatory signal, and finally cytokine production. So he is the boss. Now,
Mast cells, basophils, what are their functions?
okay, mainly they are involved in allergies and in parasitic infection, and I'll talk about detail about allergy in type 1 and type 4 hypersensitivity, type 2, type 3. So you will get to learn a lot about these in that lecture, inshallah. So everything is linked. It's just, coming slowly and gradually. Here I will just mention now, so the next lecture is about detail about Th1, Th2, Th17. Today what you should remember is that T regulatory, it is going to regulate the immune response. Okay, so they are going to suppress the tumor immunity and sometimes extra immunity, that is, more immune response that is happening around it. Th1, they promote immune response to tumors and also to intracellular pathogens. So when you want to kill intracellular pathogen, you should have Th1 active. Then if you want to kill extracellular pathogen, you should have Th2 active. Then sometimes in certain extracellular bacteria and autoimmunity, Th17 is involved. What they do, inshallah, next lecture. Now, two small things before I finish. Natural killer cell, they are called as naturally, they are able to kill the cells. Which cells? The cells that have got viruses and the cells that have tumor in them. Just look at this figure, that they can kill the bacteria, intracellular bacteria or the cancer cells, and they are part of the innate immune response. And the last thing is about the gamma delta T cell, which are just the 5 to 10% of the immune response. They are more part of the innate immune system. Why? Because they do not have MHC and MHC class 1 or 2 for their function, and they are mostly functional in the epithelium of the gastrointestinal tract. Any questions?